Good afternoon, everyone. I'm Ben Butkus, Editorial Director for GenomeWeb, and I'll be your moderator today. Today's webinar is entitled Enabling Non-Model Organism Genomics with Multidimensional NGS Datatype. The sponsor of this webinar is Dovetail Genomics. Our presenters today are Adam Abdul, Director of Product Management at Dovetail Genomics, and Ellie Armstrong, a PhD student in the Petrov and Hadley Laboratories in the Department of Biology at Stanford University. A reminder to attendees, you may type in a question at any time during the webinar. You can do this through the control panel, which usually appears on the right side of your screen. Click on the Q&A box in the upper right side of the control panel. When you click on Send To, please select All Panelists. We will ask the panelists questions after their presentations. We'll now begin the webinar with Adam Abdul of Dovetail Genomics. Please go ahead, Adam.
Thank you, Ben, for that introduction, and thank you to GenomeWeb for hosting this webinar. Before we start with the presentation from Ellie, we'd like to do a brief introduction to the technologies, specifically around proximity ligation, which help enable Ellie's research and researchers around the globe. So starting here from a very conceptual level, we'd like to set the groundwork for how this technology works and why the data is truly multidimensional. So on the right, we see two schematics representing both traditional NGS sequencing at the top, and on the bottom, we see a schematic representing proximity ligation data type. When we think of NGS sequencing and sequencing in general, we typically think of DNA in a linear fashion. And the reason we think of it this way is that, in fact, we do treat DNA as a linear molecule. We process the DNA from chromatin, getting fragments of DNA that are linear, and go about sequencing based on the technology. So what this ends up producing is data that is in a linear fashion. And the real differences between the data types are really what the size and distribution of the inserts are. So for shotgun and Illumina-type libraries, we'll have smaller inserts that are distributed across the sections of the regions that are targeted, or in the case of whole genome, across the genome. For certain other long read, linked read, single molecule type data types, we have longer reads, but still forming a linear DNA molecule and mapping to that molecule. With proximity ligation, we're actually not starting with linear DNA, and that is the starting point which differentiates this technology. In fact, what we end up having with this distribution is not a linear distribution, but we have more read separations from regions of the DNA that are in close proximity on the genome, which is pictured at the bottom in that first row. And as the read separations, or as the genomic distances increase, we have less contacts that map to that read separation. And this distribution holds true throughout the entire genome. So this distribution that we see here actually provides another dimension and a spatial component to the NGS data, which empowers a lot of different applications, which we'll talk about shortly. So briefly, how is the molecular biology able to produce this data type? At Dovetail Genomics, we have three different molecular techniques. With Chicago molecular technique, we start with high molecular weight DNA, which is reconstituted back into chromatin. And this has very specific applications around genome assembly, especially for correcting order and orientation and increasing the contiguity of genome assemblies. The second technique is the FFPE technique, and this is specifically designed for the processing and analysis of FFPE samples. And the third technique is the Dovetail Hi-C technique, and this starts with endogenous chromatin in the form of a raw sample. With these techniques, the first starting point is the fixation and locking in the conformation of the actual chromatin and the DNA. So in the case of FFPE, the chromatin is already locked and fixed into conformation in the fixation process. for both Chicago and Dovetail High-C, this is the starting point. After your chromatin is fixed, the DNA is then cut. After the DNA is cut, there's a processing that occurs, including biotinylation and cleanup of the ends of the DNA, followed by a ligation step, creating chimeric molecules. These chimeric molecules range from short separation distances out to the length of the input DNA. So in the case of endogenous chromatin in FFPE, this is the length of the whole chromosome. The fact that there are more likelihood of two pieces of DNA or two fragments being in genomic proximity and in actual proximity is a lot higher than fragments that are not in close genomic proximity being in close spatial proximity is what creates the distribution that we saw initially. After these chimeric molecules are created, then they're actually processed with Illumina sequencing chemistry and sequenced. The sequencing data is then placed into application-specific software. So in the case of scaffolding, that software at Dovetail Genomics is called highrise. And in the case of structural variation, which we will touch on these a little bit later, that software is called Selva. So what does this multidimensional data type empower, or what challenges does it help solve? So we're just going to touch on a couple applications here. Ellie will be talking specifically about genome assembly and how the dovetail proximity ligation techniques and software are able to empower her downstream research. But we'll also touch on a couple others here. Starting with structural variation, which has been a challenge for NGS. And this is for several reasons, including the fact that many of these are complex and have multiple breakpoints, and the distances between those breakpoints can be extremely long. There are also large repetitive regions around a lot of these large structural variants. And then lastly, in fragmented FFPE, the DNA is not conducive for being able to span the distances needed to be able to identify and resolve these variants. So on the right, you can see an image of this distribution that we introduced conceptually at the beginning, where along the axis, the x-axis, you can see that, in fact, most of the read contacts that you see occur close to the axis, meaning that they are in close proximity to each other. When you have contacts that are occurring substantially off the axis, this is indicative, in the case of FFPE and structural variation, of a structural variant. So there you could see an image, a zoom in of a region of this whole genome sequencing analysis. And there you could see densities of contacts that range both starting at the breakpoints, which are at the corners that you can see here, and extending the length of the whole structural variant. So this does a couple of things. One, it both is able to give lots of reads that span the breakpoint, starting at read pairs that span close to the breakpoint and the breakpoint itself, all the way to reads spanning the whole length of the structural variant. So this helps us both in identifying and resolving the structural variant, no matter how complex it is. So in this case, you can see a rather complex variant occurring between chromosome 3 and chromosome 6. And to remind everyone, this is actually from FFPE tissue. Another application that is enabled by proximity ligation dovetail solutions is for genome assembly. And the reason that this can be challenging is that in some cases, the distances between scaffolds is too great and cannot be crossed or connected by traditional sequencing technologies. Or in some cases, there's insufficient data or the assemblers make errors when they stitch the initial draft assembly together. So the assembly may be incorrect order and orientation for certain segments. So with both our Chicago, which is used for more fine resolution and correcting order and orientation and increasing contiguity, and then with Dovetail High-C with highrise, that's able to scaffold up to whole chromosomes, we're able to get greatly increased order and orientation accuracy as well as contiguity. And so the image on the right is showing an example of an output, this matrix output of a genome assembly out of highrise, High-C with highrise. And here you can see all the contacts along the diagonal, close to the diagonal, meaning this is in the correct order and orientation. And we can see here 9 super scaffolds, which are representing of the 9 chromosomes of the lettuce. And lastly, there's an emerging field that's quite exciting that has a great application for this technology as well. That is gene regulation. So previously, many of the QTL models really relied on the proximity of some of these enhancer promoter elements in order to identify those relationships. It turns out that that is insufficient for a lot of these relationships which occur at distances that are not proximity. So there are these genomic features that occur within the chromatin that actually place these enhancer promoters in proximity with each other through these three-dimensional contacts, which are also able to be profiled with high-C data. So on the contact map, you can see these triangles along the diagonal, which represent these areas of interaction within the chromatin that are not in linear space, but in three-dimensional space. And on the right, you can see a schematic of what these look like. And these particular features are called TADs, or topologically associated domains. And there's a lot of great research showing that these TAD, these looping structures with the DNA within them being able to interact, are extremely important for gene regulation. And so lots of great research occurring there, both in genetics and in terms of disease. So with that, I will wrap it up and turn back over to Ben and to Ellie for being able to talk specifically about the downstream research and the exciting research that she's doing. Thank you.
Thank you, Adam. A reminder to attendees, you may type in a question at any time during the webinar. You can do this through the control panel, which usually appears on the right side of your screen. We'll now continue the webinar with Ellie Armstrong of Stanford University. Please go ahead, Ellie.
Thank you so much, Ben and Adam, for the introduction to the technology, and thanks to everyone for being here today. Like they said, my name is Ellie. I'm a second-year PhD student at Stanford. Today I'm going to be talking about how we can enable non-model genomics through de novo genome assembly, specifically through assembling high-quality de novo genomes. I'm going to cover some of the challenges that we face when assembling non-model organism genomes and what impressions I have gotten from others about what are the most difficult aspects of assembling a genome de novo, and then I will cover several projects which have used Dovetail technology specifically to overcome some of these common challenges. So I'll first start off with a question that I'll just sort of put out there that you can think about. When I was first asked to give this talk, I actually did some research on the history of kind of the non-model genome field. So this is a little bit of a trivia question. So does anyone know, or can you think of what the first non-model organism assemblies, or what were the first non-model organisms to have a reference assembly? And I will specify that by non-model, I mean no agriculture or domestic, so no cows or corn or cats. So the answer is actually the platypus. So hopefully some of you were able to think back about what that was. And a close runner-up behind that was actually the giant panda. So the platypus assembly was published in 2008, and the panda followed shortly afterwards in 2010. So these are really great, obviously charismatic non-model organisms that were first published. And just using these two genome assemblies as an example to sort of walk through some of the statistics that I'll be discussing later about continuity and assembly quality. If we look at some of these, so the platypus and the giant panda have genome sizes of about 1.84 gigabases and 3.2 gigabases respectively. So this is well within what we would kind of expect for a normal mammalian genome. And the scaffold N50, which is a common kind of assembly statistic of continuity, which represents the scaffold size at which 50% of the total genome size is covered. So the platypus was 983 kilobases, and the giant panda was 1.2 megabases. The contig N50 for each of these assemblies was 11 kb and 40 kb for the platypus and giant panda respectively. And what you're looking for with sort of a contig N50 is that it's at least as long as what you would expect the average gene size to be within that organism, so that you have a continuous stretch of DNA that does not contain any ends or gaps covering your complete genes. So just looking at these in themselves, these are great assembly statistics. However, if we look at the methods that were used to develop these genomes, you can see that they're extensive. So for the platypus, they used a combination of plasmid, phosmid, back libraries, as well as fish to kind of align those larger scaffolds to the different chromosomes. And with the giant panda, they used 37 paired end sequencing libraries. That's a lot of paired end sequencing libraries. That's a lot of time. That's a lot of panda, most likely, and it's a lot of effort kind of with the downstream analysis. So kind of thinking about that and using that as a starting point, what are some of the common challenges that we face using when assembling non-model organism genomes? And I will say that some of these challenges certainly you come across if you are going to be working with a model system, but I think that there are particular parts of them that are more challenging when you're sequencing a genome that has never been sequenced before. So the first and most obvious challenge that we have with any genome assembly is that there is uncertainty in genome size and or complexity. And so without sequencing a genome, you mostly have to make an educated guess about the size and complexity, which can come from either previously sequenced genomes or genome size estimates using something like flow cytometry, which would give you something like a C value. And so this graph on the right, which is from the Animal Genome Size database, which if you haven't checked out, I definitely encourage you to. They have gone through and collected from the literature all of these different kind of assembly estimate values, either from directly from the sequencing data or from these flow cytometry values. But even with these estimates, you can see that it is difficult to guess what the actual genome size of your organism may be because it's not necessarily conserved within taxa. So if you plot these different groups like so, you can see that if you're going to assemble something like a bird, you can have a very predictable assembly that'll be somewhere between about 900 megabases up to about 1.2 gigabases because the genomes of that group are conserved. However, if you're going to look to sequence something like an amphibian or a plant, you might be getting yourself into more tricky territory. Furthermore, for the most part, genome size is not a great estimator of complexity. Of course, if you're getting up into the really, really giant genomes, you can pretty much assume that you're going to have highly complex and repetitive regions. But even with small genomes, what we found is that there are still regions that are highly complex and difficult to assemble, as well as whole genome duplications, and that can cause difficulty downstream. So these values are not necessarily a predictor of how difficult our assembly will actually be. So the next challenge with non-model genomes is that of sample or tissue availability and the preservation of those tissues. So the amount of high molecular weight DNA that needs to be acquired for some of the library preparation methods that you use as the input to generate your read data can be a limiting factor for multiple reasons, whether that's the sheer volume required to obtain that much high molecular weight DNA or keeping the DNA preserved preserved enough to extract the high molecular weight DNA. So for non-model organisms, this can be particularly challenging if there's regulations surrounding the samples, such as CITES. You can imagine that importing these samples and having them sit in a situation like customs where the they're not able to be kept cold or the buffers are maybe not reliable. This can cause delays and the DNA can degrade significantly over time, as well as if you are working with an organism that is particularly small, you may not be able to get enough material out of a single individual without causing significant stress to the animal, which is, I think, something that people are decently concerned with to generate the amount of input DNA required. And if you're working with a species where it's not possible to generate something like an inbred line, the more individuals you add to kind of your pool of sequence reads that you generate can cause downstream issues because that impacts the heterozygosity of the assembly and thus how well the assemblers are able to work. So the third one is the cost. So depending on the size and complexity, it can take a lot of time and resources to complete these projects. And I say this specifically for the non-model organisms because often people are now using, kind of in this big data era, are using genomics as a tool in a toolkit. So it's one of many tools that they're using. And if you're dividing up your research kind of between field work or conservation aspects as well as the genetics on top of that, and you're using genomics as a tool to enable that research, this can get tricky pretty fast, especially if you don't know what you're getting yourself into because of size or complexity estimates. So for example, researchers just assembled the Mexican axolotl. So this is this Mexican salamander. Its genome is 32 gigabases, and it has a large number of repetitive sequences. While this is really great, and I think will provide interesting information for kind of some of these larger genomes and what they look like at a large scale, if you had started an assembly project thinking that you were dealing with a smaller kind of lizard genome or salamander genome and ended up with this, you would definitely be running into some roadblocks. And then the last challenge for non-model genomes is that of bioinformatic expertise and or computational limits. So for many assemblers, along with the sheer amount of data that you will actually create to generate a downstream de novo assembly, this will obviously depend on the size and complexity, you may need huge amounts of memory and disk space that are not necessarily readily available to universities or institutes that don't have kind of an internal computing structure. More so, there are so many assemblers that are out now, it can be difficult to figure out which one to use to generate all of those assemblies and compare them. And evaluating which one is right for your data can be difficult, as well as filtering that assembly afterwards to understand which assembly is actually your best assembly. So those are the kind of the four major challenges that when I've worked with other groups on different non-model genomes that we have kind of faced. So to transition into how Dovetail can kind of circumvent some of these issues and what are the benefits. So on the right side of your screen, you'll see some of the different services that Dovetail offers. So they can produce a starting assembly, and as Adam discussed, you can go with the Chicago library prep method or the Hi-C service. What we have found that has been really great for non-model organisms is that Dovetail, compared to other methods such as PacBio or Nanopore, has relatively low input, although you do need the high molecular weight DNA. For us, this has required about 500 nanograms of DNA. That's 500 microliters of blood if we're talking about mammals or also small arthropods. I'll kind of discuss these specifics a little bit more later when I go over the projects. It's affordable for many organisms. Dovetail just released a new kit. If you haven't seen this, I encourage you to go check it out because I think that'll be really exciting. And the assembly services are provided if you so choose. So that kind of circumvents the you kind of banging your head against producing the most perfect or best assembly because they'll assist you all along this process. And as you'll see from the different projects that I'm going to start going through, they all have generated different results. But each one of the services on its own has always vastly improved the assembly, regardless of kind of the taxa that we've been working with. So the first project that I'll be talking about is set in Hawaii. So it involves a genus of spiders called Tetranatha, which are commonly known as long-jawed spiders. In the continental U.S., they kind of are known for building their webs over water. But when they reached Hawaii, they changed up their ecology a little bit and became, half the clade became cursorial predators. So they run around kind of trying to find prey on the ground and also kind of adaptively radiated across the islands into multiple different ecotypes. So for those of you who are unfamiliar with the biogeography of Hawaii, the landscape is actually age-structured and forms a really nice natural setup for questions on ecology and evolution. So if we look at this map, you can see that Kauai is the oldest island. It's about 5 million years old, and Hawaii is the youngest island at about its oldest point, 500,000 years, as well as with currently active lava flows. It was a very exciting place to work at the time with lava popping up randomly. And when we've sequenced the different groups that have colonized the Hawaiian Islands, as you can see in this bottom panel, which pictures the phylogeny of the picture-winged Drosophila, which is pretty much a textbook example of an adaptive radiation, what we found is that there's kind of this island hopping model where the species has come in from the outside, it colonized Kauai, and then it sort of just hopped down the island, speciating as it goes in this very predictable kind of fashion. And so the spiders, the Tetranatha spiders, are not an exception to this rule. So they also have kind of moved down the islands in this island hopping manner. And if you look at this phylogeny on the left, what we actually found has has found happened, which was discovered by my old advisor, Rosie Gillespie at UC Berkeley, is that these spiders have kind of, in a parallel evolution fashion, re-evolved these different ecotypes multiple times across the islands independently. So in the top box, which is Kauai, these are our two oldest species, Tetranatha kauaiensis, which I'll be focusing on today, and Tetranatha pilosa. And you can see these different ecotypes on the panel in the right. And so they've sort of moved down the islands, as you can see, to Oahu and to Maui Nui, re-evolving these different colors and different species as they have gone along. This kind of color change that happens in Tetranatha kauaiensis is also very interesting. So the spider as a juvenile actually starts out as this kind of bright green morph, and then eventually as an adult transitions into maroon. And we find that this color change actually stabilized on the younger islands, as you can see in Maui Nui and on Oahu, when there are just kind of these single color spiders. So this is a really interesting example, and we kind of had some questions about how this trait evolved, whether we could track this evolution through the genome. So we wanted to look at whether the same families or genes were recruited for color across the islands, if the genes responsible for color were originating from standing variation and novel mutation, and what genes were responsible for long-term color or developmental color change. And on the left panel, you can see the kind of different morphs of Tetranatha kauaiensis, starting in A as a juvenile, B as a subadult, and C as an adult spider. And what's important to note about the Hawaiian Islands is that all of this evolution has happened very rapidly, obviously across the last 5 million years, generating many, many species over a relatively short period of time. And what people have found looking at genomes previously is that often kind of large-scale rearrangements or inversions are responsible. There's lots of kind of synteny changes over time, and so this is something that we wanted to keep in mind when investigating these questions. So we started with sequencing the spider with 2 Illumina libraries. We had a paired end and a mate-paired, non-size selected mate-paired library from Illumina. We used the AllPass-LG software from the Broad to assemble our starting assembly for input into our next Dovetail assembly. We used the Chicago technology and then completed a high-rise assembly. And it's important to note that for the Chicago and the Hi-C methods, they do have different starting requirements for your assemblies, which is listed on that panel that I showed previously. However, Dovetail is definitely willing to work with you on whatever your input assembly is to make it kind of get the most bang for your buck and get the most out of this technology that you can. So here are some of the statistics from all of the currently published spider genomes, as far as I can tell, or as far as I've found. And what's important to note about spiders is that they can be particularly tricky. So they're kind of one of those groups that if we were to look at that kind of size and complexity graph, they would be all over the place. So the tarantula has a huge genome. It's almost 8 gigabases. Some of the genomes are much smaller, around 1 gig, and then there's kind of everything in between. In addition to this, they have a high AT content as well as many complex repetitive regions because of their silken venom genes. So getting a high-quality assembly was something that we really wanted to ensure to be able to look at these syntenic changes that have possibly happened across the evolution of this group. So on the top, we'll just focus on the 2 assemblies on the top, which I've highlighted in bold. So Tetranatha kauaiensis, for the Broad AllPass-LG assembly, we started with about 14,000 scaffolds and a scaffold N50 of 281 kilobases. And with Dovetail, we were able to improve this to about 1.9 megabases and more than half the number of scaffolds. So we saw a vast improvement in the continuity of the assembly, not so much an improvement in the contig N50, which is what we expected using this technology, but a great stride was made in at least the continuity of the 2 assemblies. And I'll also just highlight the house spider. This was done by a different group, and I believe the paper is now out in BMC Genomics, but there's also a preprint on bioRxiv. The house spider also was assembled using Dovetail technology, and you can see that they also had a very, very large scaffold N50 and a couple more scaffolds just because of their input assembly, but also just to note note that across these groups, these are some of the best spider assemblies that have come out, and this is very much for a group which people had struggled with sequencing genomes previously. So continuity is not the only way to assess whether or not an assembly has high quality. Another measure of this is to search kind of these conserved gene groups. We did this with a program known as BOOSCO. There's also a former program that's no longer upkept called STAGMA, which you can use either of. STAGMA only has eukaryotic genes, whereas BOOSCO has kind of these more specific groups. So this is the arthropod gene set. So what it's doing is it's basically looking for these highly conserved genes across the assembly and querying whether they're on kind of in a complete set on a single scaffold, whether they're duplicated and found in multiple scaffolds, fragmented across multiple scaffolds, or missing entirely. So again, just focusing on the 3 assemblies that we were looking at, the all-pass LG assembly, the dovetail assembly, and then also the house spider, just for reference. And just from the all-pass to the dovetail, we actually increased our BOOSCO scores. So we found more complete BOOSCOs. We found more single copy ones, indicating that this was actually joining scaffolds or resolving scaffolds that were maybe misassembled or not together before. We reduced our number of duplicates and reduced our number of missing genes overall. And the house spider also has really great BOOSCO scores, indicating that it is also a high quality and continuous assembly. So for the spider, which is kind of considered to be one of like a more difficult assembly category in terms of complexity, overall we've done really well. We're still analyzing this data, so stay tuned for updates on that project. So the next project I'm going to talk about is on understanding inbreeding and diversity declines in the lion. I'm really excited to share some of these results because we just got them back next week and we're really excited about them. So this project is one of my main focuses of my PhD. So as I'm sure pretty much everyone listening is well aware, is that many of our charismatic megafauna are kind of in trouble. There's been sharp declines in lion populations due to primarily habitat loss, as you can see in this graph on the left. This is their historic distribution, and then their present distribution is highlighted in blue. There's also issues with the human-wildlife conflict as kind of human cities expand into some of these natural areas, as well as the illegal trade. So some of you have probably heard about the lion bone trade in the news. To make lion bone wine kind of in place of tiger bone wine. So lions are kind of being hit from every aspect. And in the last 100 years in particular, and the last actually 10 to 20 years, their populations have declined rather sharply. So we went ahead and sequenced the lion genome because we wanted to kind of understand more about these inbreeding and diversity declines. Our starting genome for the lion was built with 10x using the 10x genomics chromium technology. You can see the assembly statistics there on the left in the table. So we had a scaffold N50 of about 10.45 megabases, and that was composed of 66 scaffolds. We had 8,000 scaffolds, and the total genome size is 2.4, so again, kind of well within that mammalian genome size range. And with Dovetail, we were really happy to say that our scaffold L90 and N90, so the number of scaffolds that compose about 90% of the entire genome is 18. So we were able to reduce that from 243 to 18 scaffolds. Why this is so exciting is that cats actually have 19 pairs of chromosomes, so 18 autosomes and 1 set of sex chromosomes. So it looks like we are pretty much approaching the chromosome level with this assembly, and you can see that link density histogram on the right where the mate pairs, as Adam had discussed, have been plotted on the X and Y axes, and there's just kind of this beautiful central pattern streaking across there. So this is really exciting for us. But I think a question that many people have is, why do you need such a great assembly? So what's the point? Like if we've assembled the gene regions, what is the point of improving our assembly further? Which is what a lot of people ask me. So for this specific project, I think that there's a lot of applications that can be used for a number of different non-model systems. Specifically for the lion, there's been a lot of talk around what are the genomic landscape changes associated with rapid population declines, and what makes some of these declines so bad for populations versus populations who do relatively well during these declines or during these boom and bust periods. So in 2015, DeBrinen et al. Published a cheetah on the paper. It's a great paper. I highly encourage you to read it if you haven't. And they showed kind of these different heterozygosity or SNV values across a number of different taxa, and that the cheetah had this really low heterozygosity. The cheetah has kind of a unique genetic population history. It went through a major decline about 10 to 12,000 years ago, I believe. So it was kind of set up with kind of a lower heterozygosity rate than normal, and then with human impact kind of crashing those populations additionally has caused additional stressors, and they're known to have reduced fertility. Then in 2016, a paper was published by Robinson et al. On the island fox, and what they did was they compared the heterozygosity across the island fox as compared to the mainland gray fox. What they showed was that despite very, very low heterozygosity, there seemed to be no population level impacts. And then in 2017, a paper was published by Rogers et al. Showing that for the Wrangel Island mammoths, which was kind of this island population of mammoths off the coast compared to the mainland mammoths, that there was this enhancement of deletions and insertions and these swapping and increased stop codons. So the questions kind of surrounding these issues will require a much finer lens of being able to track maybe these copy number variants or structural variants across time, across populations to understand what's really going on and whether or not there's a pattern with these populations that experience these increased declines or kind of this genomic meltdown, as some people have termed it. So we're really excited to use that assembly, and again, stay tuned for updates. So the last project that I will discuss is actually, I am sharing it for a colleague of mine, Katie Grbeck. She was kind enough to let me speak about her work today. Katie studies the 13-lined ground squirrel, and she studies hibernation biology. And hibernation is a really interesting phenotype that I think not many researchers have looked into, and we don't understand very well. So although they don't have a model organism, they sort of want to use this 13-line ground squirrel as the model hibernator. And this is the ground squirrel pictured in these 4 panels on the left. It's pretty cute, as you can see, so another nice charismatic organism, not as charismatic as the spider, in my opinion. But the 13-line ground squirrel has a really wide range across North America, which you can see on the right panel there. So hibernation is an adaptive strategy. There we go, sorry. Hibernation is an adaptive strategy to survive these kind of long, cold winters when resources are scarce and temperatures are low. So what the squirrels will do is they enter into this energy-saving state called torpor that's characterized by a reduction in the number of physiological processes that I've listed across the top here. So metabolic rate, heart rate, respiratory rate, and body temperature will all drop for the squirrel during this period of time. And so what you can see in these areas that I've highlighted here, during the spring and summer months, the squirrel is just kind of acting as a normal mammal. It has a normal body temperature. It emerges, you can see in the early spring, as this nice skinny fit squirrel. It starts eating, and actually by September, which you can see at the end here, it's just about doubled its body mass. So once it does that, it will start entering into torpor, which is this kind of yellow part of the graph I've shown here, with where this decline of these physiological processes begins to happen. And then in the blue, they'll enter hibernation, which include these kind of oscillations. So they spend most of their time in torpor to conserve energy, and they subsist solely off of their fast reserve. The torpor bouts, which you can see are the bottom parts of the graph, last about 1 to 3 weeks, and then they will spontaneously re-arouse to euthermia, which is something that I think the researchers don't understand very well, but is something that's obviously needed to maintain homeostasis. They spend 12 hours in this euthermic state, so these are the peaks of the graph during these winter months, where they kind of restabilize their heart rate, their respiratory rate, and their metabolic rate. And then go back into torpor. So hibernation is really composed of these cycles between torpor and arousal, and it's a circa-annual rhythm for most organisms that undergo hibernation. So it happens every year regardless. However, because hibernation is a phenotype, there are differences across organisms, individuals, and populations. So what Katie's group wanted to do was to provide this high-quality genomic resource for the researchers working on the 13-line ground squirrel. So similar to other systems that have built up their genomic resources, such as like with the domestic dog, there was just a new domestic cat genome released. The human has obviously a ton of resources thrown at it. So they wanted to really establish a genetics framework for being able to study these phenotypes and better understand hibernation. So we'll talk a little bit about how they use Dovetail and why specifically with this assembly, having an assembly that is this well put together, continuous and accurate was really meaningful for them. Their specific question for this project was that they wanted to identify the genetic variants that were associated with the seasonal timing of hibernation onset. So that was that yellow kind of portion I showed on the graph in the previous slide. So it's when this torpor is onset. So when this process starts is, again, different across individuals and populations. So they started with a genome that was produced by the Broad in 2011, which you can see in this, the statistics for in the last panel here, and then the approximate fold improvement across the bottom. And for this assembly, they used both Chicago and Hi-C, which is commonly used by many researchers depending on their starting assembly. So as you can see, across all of the assemblies, they reduced their number of scaffolds from 12,000 to about 7,000, and the number of scaffolds in the N50 went from 80 to 33 to 6. So their draft assembly includes 539 scaffolds, and their new N50 was 33 for the Chicago and then 6 for the Hi-C assembly. So that's a 24-fold improvement from the draft assembly. So the resulting assembly after Chicago and Hi-C really is practically at the chromosome level, which I think is pretty much invaluable to this kind of research. Whoops, there we go. So what they did was they did GWAS.
Please stand by. We are experiencing technical difficulties. We'll now enter the Q&A portion of the webinar. Our apologies, we're experiencing some technical difficulties for a moment. The first question is for Adam. Adam, with such a large volume of human clinical trials that sequester FFPE tissue, what is the outlook for Dovetail to enable use of its proximity ligation technology in clinical trials?
Thank you, Ben. That's a great question. So absolutely, there's tremendous value in being able to analyze FFPE samples that previously were not able to be analyzed for some of these large structural variants, which could be prospective great biomarkers for targeted therapies or even untargeted therapies, immunotherapies. So right now, our current position is this is a discovery tool, absolutely for biomarker discovery and moving into clinical and translational research. So yeah, absolutely, there's great value for many different segments, including pharma and other clinical researchers with FFPE samples that previously these structural variants, which could be very clinically valuable, have not been able to be assessed in the correct manner.
Thank you, Adam. The next question, how does the assembly software account for differences between data sets?
Thank you. I think I'll go ahead and take that, Ben. That is also a great question. And we didn't have time in the introduction to talk specifically about how that distribution pattern that we introduced conceptually is specifically used for the assembly process. The highrise algorithm actually creates a custom statistical model for every data set and looking at the specific distribution of that data set. And even so much as after certain breaks and rejoins are made in the assembly, the statistical model is actually updated with a new model to increase the confidence that the next change or scaffold would actually be at a higher confidence. So absolutely, the model is unique to every single sample and data set, and even unique between every iteration of the run of the software.
Thank you, Adam. Ellie, are you able to hear us at this time?
I can hear you. Can you hear me?
We can hear you. Thank you.
Oh, great. Okay. I'm sorry about that, guys.
Ellie, is there anything else you wanted to add to your presentation? You were nearing the end of it.
Oh, yeah. So I just, so I guess just to wrap up Katie's work is that for GWAS or something like GWAS, an analysis like GWAS, I could just go back to these slides quickly. It was really, really great for Katie and her colleagues to have these, so fewer scaffolds to kind of find these outlier variants and kind of reduce the number of areas that they were looking at. And they were actually able to find a gene or a kind of outlier variant that was located near the CHRM2 gene, which is the SNP number 4 that you can see on your screen. And that receptor is involved in reducing heart rate. And the only reason that they were actually able to find that is because it was actually in a region that was surrounding the scaffold. It's not directly on the variant, but it's directly to the right. And that scaffold was not continuous in their starting assembly, but was continuous in the dovetail assembly. And they were able to kind of locate that region and isolate it because of the highly continuous assembly. So that's all that I had to add. In summary, it's important to have continuous assemblies, and thanks to everyone for listening and all of my funding and collaborators. And that's it. Sorry, guys.
Okay, thank you, Ellie. We'll now continue with the Q&A portion of the webinar. The next question is for Ellie. When you scaffold normally using paired ends, there is physical evidence of linkage. Does Chicago/Hi-C provide direct physical linkage between scaffolds, or is it a statistical probability based on the density of linked regions?
Yeah, so this is a good question, and I think Adam can also speak to this. So if you look at the the linkage maps that we showed earlier, these link density histograms. So the way that the Hi-C and Chicago work is that you have some sort of input size molecule, and the mate pair, it's sort of like a mate pair library that the two reads are on the ends of these. So there is physical linkage usually between these regions because they come from a single molecule, but by producing those many mate paired reads, you actually are able to kind of confirm that those two reads have come from the same molecule. And then if there's any kind of issues with this, which are kind of these breaks in the assembly, you should be able to theoretically see those in the assembly graph. And if there's anything Adam wants to add.
No, Ellie, that's a great explanation. Yeah, so there is these are connected through this ligation process. And so the distribution is extremely indicative of the proximity through the statistical model of that relationship. And it's through that distribution, as we mentioned earlier, that we're able to actually provide this this ability to connect scaffolds at distances and also for the correct order and orientation. But the actual read, as Ellie mentioned, really acts like a mate pair read in many ways.
Thank you. The next question is for Adam. Is highrise available to use for a second round if we improve our input assembly?
Absolutely. So that really is where highrise shines, right? So Dovetail and/or any researcher could produce the initial de novo assembly using almost any method or technology out there or assembler out there to get that initial draft assembly. Once the draft assembly, either created by Dovetail or by the researcher, is completed, that's really the input that highrise will use to scaffold, along with obviously the Chicago or the Hi-C data, right? So absolutely, if you have a draft assembly, depending on the shape and how the contiguity of the draft assembly would determine the exact library preparation or the proximity ligation technique that we would employ, but along with highrise for the scaffolding to increase both the order and orientation accuracy, and then obviously the contiguity of the assembly.
Thank you. The next question is for Ellie. Does Dovetail just order and orient the scaffolds like FISH does, or does it provide more information than that, like how much sequence is missing between scaffolds?
Yeah, so this is similar to, I think, the question before, which is that because of the way, so you can think of it as similar, again, to mate pairs, because we have information on the molecule and how the molecules are connected to one another and which reads come from which molecules, there is actually orientation and information about the gaps in the assembly. And for Chicago, that's kind of a more predictable molecule length, and Adam can speak to this as well. And for Hi-C, those will be different because it's kind of the biological information of the folding of the molecule.
Thank you. The next question is for Adam. Are there other sequencing platforms that work well with Dovetail technology, or is there a special need for Illumina?
Yeah, and so to be clear, in terms of the draft assembly, for using as the draft assembly for being able to scaffold using Dovetail technologies and highrise, those can be produced with almost any technology and assembler. So this could be PacBio, Illumina, almost any technology out there. However, for the particular technique, in terms of the Dovetail, Hi-C, or Chicago, those are libraries that are currently set up for Illumina sequencing. So these are paired-end libraries that are run on almost any Illumina sequencer.
Thank you. The next question is also for Adam. Is there an advantage of using proximity ligation compared to arrays for structural variants?
Wow, okay, yeah, that is actually a great question. So one of the things that the proximity ligation technology is able to do is, in terms of structural variation, it's able to detect, identify, and resolve variants across all classes. So this includes balanced and unbalanced. For arrays, there's a couple things that come to mind. One, that they typically arrays are for unfixed material. I think there are a couple examples of arrays that are used on fixed tissue, but for the most part, they're for unfixed and fresh material. And they are really set up for CNVs, so really for copy number variations, so deletions, amplifications. So for balanced events, like balanced translocations or inversions and other balanced events where you don't have a copy number variation, arrays, even though they're in a whole genome in scale, will not be able to identify those variants.
Thank you. The next question is for Ellie. Based on the Lion 10x assembly, how does Dovetail/highrise integrate the 10x haplotype data, if at all?
Yeah, that's a great question, actually. So for our Lion assembly, so for those of you who are unfamiliar with how 10x works, it actually provides phased information for your genome. So when you're working with the output, unlike other kind of traditional assembly outputs, it's not a single line FASTA. You can actually output these two different single line FASTA, which more or less randomly selects the different haplotypes from the scaffolds that are phased. So for the Lion, we just provided one of the single line FASTAs for the Dovetail input assembly. I think it would be worth checking for the other one, but Dovetail does provide some Dovetail can provide phasing information itself. So if the pair, the read pair spans a heterozygous region, it will know about that. I don't know if Adam wants to kind of speak more to that.
No, Ellie. And by the way, if there are any really specific questions or continued conversations that we need, please, reach out to us. You can go to dovetailgenomics.com, because a lot of these things, in terms of custom projects or particular highly technical questions, we can definitely work through those with you. I'm sure there's many questions that a lot of the participants have. So please visit us at dovetailgenomics.com. You can fill out a form on the contact form, or send these additional questions in here, and we'll follow up with you, because, yeah, there's definitely a lot more information we can probably provide, but it would require a detailed conversation.
Thank you. The next question is for Adam. When Hi-C joins two contigs, could the orientation of the contigs be fixed?
Yeah, and I think we're using in terms of terminology. So highrise is the algorithm, the software that actually does the breaking and joining and the scaffolding. And absolutely, for both inputs around, specifically even more so around Chicago, and we're talking now about the technique, and Dovetail Hi-C, highrise not only joins scaffolds together or joins contigs together to create these larger scaffolds, but also looks at the order and orientation. So if there's either a join that already exists that does not fit the distribution that is expected, highrise will actually break that and rejoin it and test all the different iterations until it gets a join that is the consensus or the highest predictable join. And when it does this initial join of looking of different ways that scaffolds can come, or different contigs can come together, it absolutely looks at order and orientation as it tests all the different iterations. So absolutely, that is a major part of how the software operates.
Thank you. The next question is also for Adam. How can proximity ligation work with FFPE if it needs intact chromatin?
Oh, that's actually a good question, and maybe a little clarification that's needed. So the reason that it's able to work with FFPE is that the fixation that occurs in the actual tissue when it's fresh actually captures the conformation of the chromatin. So even though that breaks in the DNA are introduced, the actual conformation of the endogenous chromatin is captured. And that conformation capture is actually what's needed in order to be able to produce these libraries and then turn out this distribution, including the extremely long-range information, into the megabase range.
Thank you. That concludes the Q&A portion of the webinar. We'd like to thank our sponsor, Dovetail Genomics, and our presenters, Adam Abdul of Dovetail Genomics and Ellie Armstrong of Stanford University. If we didn't have time to get to your questions, we will try to have our panelists answer them directly afterward. As a reminder, please look out for the pop-up survey from the sponsor after you log out. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to attendees. Thank you for joining us for this genome webinar.